Biology 175 is a class called Choreographing Genomics. Now, what Choreographing Genomics is about is using a postmodern dance lens to explore genome information flow. And once the students have learned about genome information flow, they go on to explore cancer biology and the genes that are involved in having damaged genes in cancer cells. So there are two books for this class that the students use. One book is entitled DNA by James Watson, who discovered the DNA double helix. And the other book that the students use is a book called Terpsichore and Sneakers, which was written by an incredible woman named Sally Baines, who had been my professor in college. And it explores many different postmodern dance choreographers, so the students really study different modes of expression. And then they're asked to think about those modes of expression to express DNA, to express RNA, to express proteins, and everything that goes on in a cell. And that's so that they can really use their brain to think three-dimensionally about what's happening in cancer, which is very movement-oriented. And then they also sometimes translate it to what happens in the person. And so you'll get a sense here going through the different acts. So Act 1 is the introduction, me telling you about the class. And then you're going to see DNA synthesis, so the molecules of DNA, in PCR, polymerase chain reaction, which is, you hear about, you read about in the New York Times. So you'll see it, and just experience what's going on. You don't have to understand everything, but you'll just get an experience, kind of this is what's going on in the cell. And then you'll move on into DNA replication of the chromosome, transcription, and we'll just tell you a little bit about translation, which is how the proteins come off of the mRNA. We'll then move into Act 2. Act 2, I'll call out all the chromosomes. So there are 23 pairs of chromosomes in every human cell. And off these different chromosomes come proteins that are associated with cancer. So as I call out the chromosomes, you'll start to see the genes associated. So they're here, NF1, so I put them in order of tumor suppressors and then oncogenes because that's how they enter for Act 3. Each of these students, Nubia, Eli, Eunice, have choreographed, all of them, have choreographed their own pieces with relationship to the genes they chose that are damaged in tumor cells. So you'll first see them just so you get an idea of the chaos, and then they'll begin in Act 3 to actually show you their pieces of choreography, which came from seeing some dance, thinking about what they studied in Terpsichore and Sneakers, what they studied in DNA, and other readings they did.
This is our truth. This is our central dogma. From DNA to RNA to protein. DNA from itself with replication. Think cellular copying machine. You get the same thing most of the time. RNA from DNA with transcription. Think voice transcription on your iPhone. From sonic sound waves to bytes, but still in the same language. Protein from RNA with translation. Think Googling what is hungry in Italian. Different language, but same meaning. The origin of replication is a sequence on DNA that's responsible for initiation of replication. It attracts factors that bind to that sequence, which further attracts a protein called helicase, which is responsible for opening up the DNA. Have you guys ever heard the joke, you must be DNA helicase, because you make me unfit my genes?
feel, they breathe, they interact with the world around them, they move, they adapt. It's a harmonious jumble of molecules. It's all coded in deoxyribose nucleic acid, 2 strands, 4 simple bases, a sugar phosphate backbone, hydrogen bonded to one another. This is all the code for life.
Adenine.
Guanine.
Thymine. Cytosine.
PCR.
5 prime, 3 prime, 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 5 prime, 3 prime.
5 prime, 3 prime. 5 prime, 3 prime. 5 prime, 3 prime. 5 prime, 3 prime. 5 prime, 3 prime. 5 prime, 3 prime. 3 prime.
Yellow case gold. Now we'll see the process for pyrolysis. Oh, almighty central dogma. You propose a unidirectional information flow from me to RNA to protein, that I know. But I'm a visual learner, so you must show. Show me thy way, and I will grow.
Ask and thou shalt receive, young Deoxy. DNA information is essential for you to faithfully pass on your legacy. In order for me to show you, you must open up. Literally. You need helicases to help you. Now let's show you this with a rope so you can see the replication ball.
Helicase, go.
Now you are ready. First, we need primates, which is an RNA polymerase, to lay down an RNA primer on both the leading and the lagging strands. Then, DNA polymerase adds deoxynucleotides one by one, using each strand as a template.
5 prime, 3 prime. 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 5 prime, 3 prime.
There you have it, young deoxy. Now there are 2 of you.
5 prime, 3 prime. 5 prime.
Thank you so much for that information. Thanks for enlightening me, O Almighty Central Dogma. Making copies of myself is fun, but I don't want to be done. I want my information to be read, so please, show me. Go ahead.
Of course, my lamb. Sequence-specific DNA binding proteins bind to the promoter region of the gene of interest. These transcriptional factors will help recruit RNA polymerase to the transcriptional start site. This is called the initiation step. The polymerase adds ribonucleotides one by using the non-coding strand as a template. The messenger RNA is then processed in several ways and then shoved to the cytoplasm where it will be translated into protein.
Wow, translated into proteins. Thank you, Central Dogma. You've shown me the way. Now I can enjoy the rest of my day.
Yes, until mutations happen and things go astray. Yes, until mutations happen and things go astray. Yes, until mutations happen and things go astray.
Now into Act 2, where we're going to start to have mutations happen and things are going to go astray. So we're now into cancer biology. So you've seen the central dogma, and we're into the mutations in cancer genes. Chromosome 1, chromosome 2, chromosome 3,
Pick 3CA.
chromosome 4,
3CA. Pick
chromosome 5,
Pick 3CA.
chromosome 6,
3CA. Pick
chromosome 7,
Pick 3CA. Pick 3CA. Pick 3CA.
G.
Pick F 3CA.
chromosome 8,
G.
3CA.
F. R.
chromosome 9,
F. Pick
R.
3CA.
chromosome 10,
R. E. G.
chromosome 11,
R. E. G.
chromosome 12,
R. E. G.
F. Pick
R. K. G.
chromosome 13,
R. E. G. K. R. E. G.
chromosome 14,
E. G. K.
chromosome
E.
15,
G. R.
chromosome 16,
R.
chromosome 17,
R. E. K. R. E. R.
chromosome 18,
R.
chromosome 19, chromosome 20, chromosome 21, chromosome 22, chromosome 23, So that's your introduction to the chaos that ensues in cancer and all these different genes that can be expressed and all these different mutations that can occur. Now you're going to enter into the world of each individual choreographer and the particular genes that they picked. So we begin with tumor suppressors, and the first tumor suppressor you're going to learn about is NF1, which is located on chromosome 17, and this is Nubia's piece entitled NF1, It Travels.
Tumor suppression by NF1. Tumor suppression by NF1.
The sky is blue today. I woke up with light in my eyes. The day goes as it should. The world wakes up and goes to sleep. She is tired. The sky's a bit grayer, but I can handle it. Nothing is set in stone. There's always tomorrow, as long as the sun will rise. The sun came up. I stayed in bed though. Tomorrow came, but nothing's different. The world's no longer tired. She's wide awake. I've stopped drinking coffee. My hands shake so much, I don't need caffeine. My skin is starting to match the sky. Gray pores and eyes cloudy. I can feel myself fading. The world laughs at me now. My life's a knee slapper. The laughter keeps me up at night. There's no stopping her. There's no getting better. I'm tired.
So each student was asked to show you before these genes were mutated and after, so you sort of see as cancer ensues at the end. So we move now to another tumor suppressor, which is in the news a lot, BRCA1. Next piece is by Eli, and it's NF1, a change in the change, RNA editing.
In a small cell, invisible to the naked eye, lies a far-off land known as the human genome. We stand in the 17th state of this 23-chromosome nation, in the city of NF1. The city contains one street, with houses occupying only one side, and traffic moving in a one-way direction from the 5' end of the city to the 3'. Our street, however, is better known as our mRNA. Let me take you back to a time before the new family moved in and wreaked havoc on the city. It was a sunny day in NF1, and in house number 2914 lived Mr. C. He got along peacefully with his neighbors in houses number 2915 and 2916, Miss G and Miss A, respectively. You see, they shared a mailbox labeled C, G, A. And every morning, the delivery girl, Ribosome Rachel, would come through and leave an arginine package in front of the mailbox before moving on her journey. But when she got to the end of the street each day, the city would cheer. The protein was complete. Now a piece of the chain, shortly after Mr. C's house, was better known as the GTPase activating protein domain, or GAP. Now this message helped to transform the 3-branch district of GTP in the city of Ras. The message read,
Wake up, GDPAs! GDPAs, wake up! Wake up, please, GDPAs!
now one of the branches of GTP shut down, changing the district from GTP to GDP. Once transformed, the district shut down the division factory in the city of Ras, halting the production of a new duplicate world. Now let us fast forward to the day that the new family moved in, causing chaos and destruction. It was a sad day when Mr. C was asked to leave his house. He packed up his belongings, and in his place, the Yu family moved in. Of course, they changed the mailbox, and it was now labeled UGA. But when ribosome Rachel came the next morning, she halted at the mailbox, unable to move forward. The chain was left incomplete. The chain did not contain the message for the 3-branch district in the city of Ras. And therefore, NF1 could not send its message. The division factory in the city of Ras continued to work as GTP was never transformed. Each new world contained our nation, the human genome, and in the 17th state of this 20th city of NF1 still existed. And in each new world, the Yu family continued to move in to house number 2914. Each new world increased the rate of proliferation. This new mass of cells continued to divide and grow. This mass endangered the universe that we now live in, the human body.
Now on to BRCA1 on chromosome 17. Many of these are on chromosome 17, and this is by Eunice, and it's BRCA1 on DNA repair.
Inside a cell, there exist many entities to keep the peace. Tumor suppressors work, resist malignant activities that never cease. BRCA1 and P53 are 2 genes
BRCA1. P53.
2 genes coding for tumor-suppressing proteins. Inside the cell, they go repair damage to DNA that may appear. All of a sudden, tragedy does strike, and
Deletion!
BRCA1 can no longer fight. Proteins formed have no effects on DNA damage that collects. Alone, P53 is not enough to act on the damage. It's very tough. Now when the cell begins to divide, the damage follows. Oh my! Damage accumulates, cells cannot die, cancer appears, and begins to thrive.
This piece is by Shirley, and it's when BRCA1 breaks.
BRCA1 mutations increase chances of breast cancer. When normal, the gene and protein product can function and detect abnormalities of cellular DNA. Normal cells pass the test of BRCA1 and continue happily along its way. Damaged DNA is detected by BRCA1, and if working properly, BRCA1 can repair.
Repair! Repair! Repair!
A two-hit mutation of BRCA1 leads to dysfunction of the genes for mom and dad and no correct protein product. Normal DNA can go on their merry way, and damaged DNA can go undetected and continue to replicate in new cells and develop as cancer cells. Damage unite. Damaged cells proliferate, and the tumor grows uncontrollably with more and more mutations to a metastatic breast cancer.
And now we move on to P53, the tumor suppressor you've heard of already. And this is a piece by Prima, and it's entitled P53, the Key to Life and Death.
This is your DNA, out in the sun. When UV rays come, they block the fun. DNA damage calls out for repair, and MDM2 must learn how to share. Away from P53 it must go, so P53 can do its work, let us show. A normal P53 protein will call on P21 for help to stop the cell from dividing and hope the damage goes to none. If the damage is beyond repair, P53 must call upon Puma with care. Puma will call for the necessary fate of death in a cell whose destruction cannot wait. Now let's see what happens when a P53 protein is mutated at a single amino acid that makes things complicated. Now when DNA damage occurs in the cell, P53 cannot ensure that the cell will be well. It cannot bind to P21 or Puma and thus stop dividing or die. This is when things go awry. If not for the function of P53, let us ask you, where would you be?
This piece is about Puma. It's by Sarah, and it's entitled Puma, Keep It Moderato.
On a good day, cells are pretty average, guys. They grow at their own pace, not an excessively fast one.
That's because P53 and our security guards, Puma, are standing around making sure the snappy doesn't get too fast. On a bad day, a crazy jazzy cell tries to break the tempo, so P53 sends out her Pumas after him. For now, all is under control again. But on a really bad day, P53 is sabotaged. It's a mutation. When P53 is mutated, there is no one checking on the cell's tempo. No one can send out Pumas anymore. A crazy mutated jazzy cell comes around to speed up the tempo again. But Pumas are sleeping in today, and the jazz gets out of hand.
This piece is by Irene, and it's entitled Puma and the Killer Cell.
DNA. DNA. Flowing as you do. Intertwining, separating, no mutation have you. To the mitochondria, powerhouse of the cell. Sleeping and inactive, there cytochrome C dwells. Ready within a moment's notice. P53 stalks within. With DNA as its focus. There's the onus. DNA damage he's noticed. Puma is transcribed, breaking the cell's rhythm. Dancing with BCL2. Now back is free to make a schism. Within the inner membrane, where site C was laid, back creates an opening, and site C begins a tirade. Cell death has occurred. The damage does not escape. But what were to happen? Should P53 not be in its place? Scenario 2. DNA, flowing as you do, intertwining, separating, a mutation have you. To the mitochondria, powerhouse of the cell, sleeping and inactive, again cytochrome C dwells. But P53 is not there anymore. He's been mutated. In fact, he's been deleted. Who will notice then when DNA damage has been repeated? Without P53, Puma couldn't transform. The damage moves and multiplies until there's no chance for reform.
Next piece is by Stanley, and it's also Puma chromosome 19, a well-oiled machine, the Puma cat.
A properly working system is integral for the survival of the system. A broken cog can cause part of a machine to malfunction and may even cause damage to the rest of the system. That is why it is important to find the problem before it is too late. Our story starts in the nucleus. This string of parts is our DNA. It needs to be complete and pristine in order to work properly. If it becomes damaged, there needs to be some way to prevent it from malfunctioning. P53 there can recognize DNA damage and must alert Puma, another protein.
Puma!
And now to the mitochondrion. Here is where the situation is rectified. Puma binds to BCL2 to keep it from Bax. This allows Bax to release the cytochrome C to the rest of the cell and cause cell death, preventing the faulty DNA from causing cancer or other problems. And now it ends. But wait, what if one of these integral proteins is mutated? That is to say, not functioning. Here, we have a similar situation as before, but P53 cannot recognize the cell damage. In the mitochondrion, Puma is unaware of the situation, and nothing happens. The DNA is left unchecked, and the cancer has begun.
We're going to look at RB, which is on chromosome 13. And this piece is by Nadira, and it's entitled RB, Losing Sight of Control.
The RB gene harbors commands for the production of RB protein, a tumor suppressor with a vision. In its throne, RB reigns tight negative control over the cell's division cycle, allowing the cell to go through only when it's ready. G1 to S, stop to go, RB, in defined balance. With its power vested in 2 copies, 2 alleles, you will now see the story of 3 different cell fates that arise when things go awry in any one copy. G1 to S, stop to go, RB, in defined balance. RB at the edge of chaos, RB losing sight of control. In normally functioning RB, where both alleles are intact, RB keeps the cell from passing G1 to the S phase by direct union with transcription activator E2F1. G1 to S, stop to go, RB, in defined balance. Oncogenic mutations can lead to inactivation of RB. But wait, it is possible to retain the reign of order in the wake of 1 normal copy and 1 mutant copy. RB is phosphorylated and the normal cell cycle occurs. G1 to S, RB in defined balance, RB at the edge of chaos. But if light finds its meaning in the presence of darkness, does function not find its worth at the hands of dysfunction? It is in the presence of 2 mutant alleles in which total inactivation becomes a reality. Loss of RB function brings deregulation, uninhibited growth, proliferation, a call to malady. The RB gene renders the protein incapable of uniting with E2F. Cells pass from G to S, no matter the stop signals, and keep dividing, with no dignity left. Disarray floods the scene, and the monster with the power in numbers comes to play. G1 to S, RB, losing sight of control.
this and studying it in the textbooks, and it's a lot of reading. But in this class, they're doing the reading, but they're not showing you all of the reading. So here's some of the reading for you. However, if RB is overphosphorylated by a kinase such as cyclin 1, the RBE2F complex will break and unregulated cell proliferation can occur.
RB1.
E2F1. Regulate cell division.
And now we're moving to oncogenes, and this is an oncogene that's located on chromosome 18, and it's called BCL2, and the next piece is by Alex, and the title of the piece is What BCL2 Does for You.
Good morning, ladies and gentlemen. This lovely lady sitting in front of you today will represent a cell. Now, all living organisms are made of cells. Can any of you guess how many cells are in the average adult human body? Shout them out.
7.
Very close, but no. Anyone else?
5 billion.
Close. Closer. One more guess, come on.
7 billion.
People, scientists, think there are 37 trillion cells within an average adult body. So, as you can imagine, these cells need to coordinate. They need to coordinate their activities to work in unison in order to create a functional multicellular organism. Now behind, on the stage, are the events that you see occurring within a cell. Each cell has multiple genes, depicted here. Can anyone guess how many genes are within a cell? Let me give you a hint, it's much less than the number of cells in the human body.
3.
Close, but no, that's the answer. Any other guess? Closer, one more, come on. Come on guys, don't be shy. Scientists believe there are around 25,000 genes within a human cell. This is the gene that codes for the protein BCL2. That's a regulator of cell death. When the promoter region of the gene is activated, the gene turns on. After a few intermediary steps, I, BCL2, am made in my protein form. B, C, L, 2! I am BCL2, and I am currently working inside every one of you. As I said before, the cell has to be a good neighbor. It is surrounded by many other cells that constantly send it signals. Sometimes these signals are death signals. If these signals are only a few, and if the cell is healthy, I'm responsible for stopping them from killing the cell. out of here! Get out of here! Success! I have defended my healthy cell. But sometimes the cells gain mutations. In this case, the cells around this one notice and they send more death signals. Even though this seems like a violent, brutal process, it is good for the organism because we're getting rid of a bad cell. Now, in cancer, things are very different. In the type of cancer called B-cell lymphoma, which is where BCL2 gets invented, this gene is deregulated. There's a translocation event, which means one part of the chromosome gets cut off, and the gene fuses with a promoter that is always active. Therefore, you get constant production of the protein, overexpression. BCL2! Now, the cell is still mutated, so death signals try to kill it. We can block all of them because the protein is overexpressed. Therefore, this cell remains untouched. It can further divide to create cancer.
And it's called BCL2 Walk or Die.
Don't die. Don't walk, 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 walk.
Walk or die.
Walk,
Mutate.
walk, walk, walk.
Walk or die.
Walk, 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 walk. I don't want to walk. I want to read. Read.
Mutate.
Read.
Walk or die.
Read.
Mutate.
Read.
Mutate.
Walk or die.
Mutate.
Walk or die.
Walk, 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 walk.
Walk or die.
Mutate.
Walk, 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 walk.
Walk or die.
Mutate.
Walk, walk, don't die. Walk.
Walk or die.
Translocation. Chromosome
Walk or
18
die.
are moving up. Chromosome 14 is around that.
Walk or
Walk,
die.
walk, walk, walk.
Walk or die.
Walk, walk, walk.
Don't die.
I don't want to walk. I want to read.
Don't die.
Mutate.
Read.
Walk or die.
Read. Don't die. Read. Read. Don't die.
Walk or die.
Read. Don't die. Read. Read. Don't die.
Walk or die.
Read. Don't die. Read. Read. Don't die.
Walk or die.
Read. Don't die. Read. Read. Don't die.
Walk or die.
Read. Don't die. Read. Read. Don't die.
Walk or die.
Read. Don't die. Read. Read. Don't die.
Walk or die.
Read. Don't die. Read. Read. Don't die.
Walk or die.
Our next piece is another very well-known oncogene. It's about KRAS, which is on chromosome 12, and this piece is by Hannah. And it is entitled Growth Signals Gone Awry, KRAS Expression in Normal versus Cancer Cells.
On the 12th chromosome, on the 12th codon, in the reading frame of KRAS, a single base pair substitution has changed the fate of a cell forever. The 12th codon, a normal cell operates without a hitch. The 12th codon is running smoothly as the amino acids that match the mRNA attach one by one and fly off as they are added to their protein chain. The protein RAS is inactive at first. It needs GDP in order to turn on. When RAS and GDP come together, the cell receives signals to grow. Luckily, RAS can turn itself off by hydrolyzing the transformed GDP back to its original state, and the cell knows not to grow. But one mistake changes everything. The amino acids attach as usual, but the code isn't all correct, and that is enough to change the function of RAS. Mutated RAS cannot hydrolyze GTP back to its original state. So RAS stays on and growth signals continue to tell the cell to grow.
This piece is by Christina, entitled PIK3C3A, and if you can't see it, but it's PIK3 Chaos Anarchist.
The pathway, PI3K at mTOR. Many obedient components make this pathway run smoothly. This path starts with signal transduction. A ligand binds to the exterior of the tyrosine receptor kinase, activating PI3K. PI3K activates the regulatory subunit P85 to bind to catalytic subunit P110 alpha, otherwise known as PIK3CA. PIK3CA, being catalytic, causes a chemical reaction in PI3K. This allows PI3K to transfer a phosphate from ATP to PIK3 activates ACT, which further activates mTOR. ACT and mTOR stimulate cell growth and cell proliferation. P10 regulates by suppressing PI3K rebels. Cell growth, proliferation, cancer, chaos.
Now we're moving to chromosome 7 and the oncogenes on chromosome 7. And we begin with the oncogene BRAF on chromosome 7. And this piece is by Nabanita, and it's entitled BRAF, the Chaos.
When I was younger, I watched a lot of Bollywood films. There was always a clear hero, one with dashing good looks and a charming smile, who was always on a mission. There was also a villain, someone who was out to disrupt the hero's mission. In our bodies, one of our heroes is the BRAF gene, and it produces a protein also called BRAF. Our hero, like all good Bollywood heroes, has a mission, and this hero's mission is to phosphorylate. Phosphorylation! It activates proteins that are involved in cell growth and proliferation. Now, our villain in this film is the mutated BRAF protein. When a thymine nucleotide is replaced with an adenine in the BRAF gene, we get this mutated protein that results in increased phosphorylation and even more cell growth and proliferation. It's chaos. And this is our Bollywood film, BRAF the Chaos.
Our next piece is about EGFR, epidermal growth factor receptor, on chromosome 7, and this piece is by Song, and it's EGFR, not quite better, definitely faster, unfortunately stronger.
The protein product of EGFR gene is the EGFR protein, a transmembrane protein that takes an extracellular ligand and amplifies it using a single transduction pathway. On stage, you see 4 clusters of enzymes in the cluster representing cells. These 4 clusters represent EGFR, PI3K, AKT, and mTOR. This single transduction will cause cell proliferation. These are kinases which phosphorylize other enzymes. These represent phosphate groups. There are more enzymes downstream than upstream because each enzyme can phosphorylate more than once. In normal cells, EGFR takes a ligand, dimerizes, and phosphorylates the cell. Let's begin. EGFR gene, and as a result, EGFR gene protein is permanently on.
EGFR from chromosome 7. And this one is entitled EGFR Mutation Tag.
In most cases, the mutations occur in exon 19 and exon 21 of EGFR, adenocarcinoma in the lungs. I'll show you how.
I'm going to show you EGFR mutation.
Dad, you're it! What mutation on exon 19?
Tag! Frame shift mutation on exon 21.
Thank you all so much for coming and supporting us. Any questions?
No, I was just wondering, like, if you had the idea for the choreography, and then, like, sort of chose the gene because of that idea, or if it was the other way around?
I think it was the other way.
The other way around.
Yeah. When we picked the gene, we didn't know what we were picking.
That's not true. So there is a very detailed syllabus that made it clear what was going to happen during the semester. So, but they didn't, you know, during the class it emerges, and they really, I mean, in the beginning, you should ask them how much movement they do. It's like, you know, by the end, they're really big and they're moving. So they begin to really explore, so they don't quite know what they're getting into, but by the end they're all, as you can see, really seasoned, and they're moving, and they get an idea of how they need to express this through movement.
Great. Really awesome, Jen.
That was great.
Thank you.
It's good. You have a question here.
Yeah, so in the choreography book that you mentioned that you guys read, were you exposed to a lot of, like, how was it organized? Were you exposed to different types of choreographies or dance or?
Each week we had a different chapter. Each chapter was focused on one postmodern dance choreographer and a specific aspect of their work. So we focused on choreographers like Steve Paxton, Lucinda Childs, Simone Forte. We talked a little bit about Merce Cunningham's collaboration with John Cage. And so through that, we were able to learn different techniques, but mostly take away from it that postmodern dance is anything and everything that we think it is.
You have another answer.
Yeah, and we also were able to go to Fall for Dance. So we were exposed like not only through YouTube videos, but we were able to like watch performances in person. So for a few of us, that also inspired our choreographies.
You have a question?
Is there anyone majoring in dance or music? Not a single one.
Could you tell?
Yeah, most of us are actually majoring in science. So, right?
Yeah.
Most of us are majoring in science.
Psychology. I mean,
Yeah.
Kind of sciences.
Yeah.
I'm a psych major, so.
Okay.
I think, how many bio majors do we have? Two. Three? And we have freshmen,
Through freshmen through seniors.
How much rehearsal time would you have to put in? Because it seems like would have been hard to remember everybody's choreographies. Because you all did so many of them.
I mean, I feel like we didn't have a lot of rehearsal time, but we were really good at communicating what we wanted because everybody had their own piece that they were in charge of. So it was all about communication, and I feel like we had about 3 weeks to prepare. So we were, by the end, we were pretty solid.
Also, like, it was really nice that we were able to record the dances and then put them up and just share them with each other. So, like, even if we couldn't meet in person, you could watch the recording and
We
know what you have to do.
So yeah, I made a Dropbox and then invited everybody to Dropbox. So everybody was sharing everything in Dropbox. But the class itself, so they only were required to meet during class time, right? So we have a class meeting time. So they were all there and they all became a real community during the class time. But then that community had to happen outside, and that happening outside, there was a Dropbox, and then they could figure out how to communicate to. But everybody is a choreographer, everybody is a dancer. They all have to do, so they learn about every other gene.
I have a question here.
Jill, can you talk about the structure of the class? And with, how much movement, how much sitting down, lecture?
So there really is, they should talk about it too a little bit, but there really is very little lecture except when they weren't getting a concept, and then I would get a little upset and I'd be like, okay, we're gonna do this again because I got the homework, and it's, because they would have to give me homework where I could really see, are they getting this concept? They had to design PCR primers, for example. And that's a scientific concept. It's like, okay, did you get it right? Do you understand 5' to 3'? So that in the beginning they did a lot of this stuff trying to understand 5' to 3'. And if they didn't get it, I would lecture, and then I'd bring in a scientific slide. But we had no chairs in the classroom. It's an open room. So they'd just sit on the floor, and I do that. We began in the beginning of the semester, we did exercise, because just to get them understanding to move their body. So the first 20 minutes would just be movement that had nothing to do with science, just so they would move their bodies and try to learn how to do it together, be on the same time, so they'd learn about moving together and being on the same time, but not telling them that's what they were doing, but they were doing it. And so there was a bit of everything, but maybe, he doesn't.
So there wasn't much, as Dr. Barganetti said, lecturing where we were sitting down, but I think for a beginner to science, it's more important to learn the subject through movement. You get a misconception of what science is, how things actually work, but through movement. So it's really a class where you combine lecture and movement at the same time, like Dr. Barganetti was saying. We're doing the 5' 3' until it got a little bit more Exactly.
Yeah, we really did that.
So especially for people that are science beginners, I think it's a great way to learn it.
And like I feel, me personally, I didn't understand DNA replication until I took this class. Like it's just a different way of looking at it. And Professor Barganetti really was big on making sure everything was scientifically accurate, and that when we learned, visually, but she also made sure that we understood every single thing that was going on with our movements. So.
Sometimes when people would get things incorrect on the homework assignments, she could see that we didn't fully understand the concepts. So it would end up most of the time us laying on the floor for about 15 minutes with her yelling. Or, you know, you guys are now chromosomes, contact, improv, show the dividing, show where you guys are ending up, show how mitosis and meiosis works. Just like basic concepts where we were forced into this and having us pushed and pulled around the room as if we were in the cell working. So I thought that was another way to look at it that really forced us into the atmosphere.
Yeah, and if they got it wrong, they had to do it again. Because you could see that it wasn't right. And it was like, no, you wouldn't be there. Do it again. Where would you end up? And they would have to figure out where they would end up in space.
I know a lot of you have a science background. How many of you have had movement background before this class? And how many of you who have not are going to continue maybe exploring more movement and just in your life in general because of this experience?
Well, I mean, as you probably can tell, I have zero movement background. I also am not like musically inclined. I don't know how to play an instrument. None of that. But like when I saw this class, it kind of combined two things I was interested in. Like I am interested in dance. I just never tried it out because this is the best thing I can do. You know? Like, I'm sure you guys have seen that's my best work. So it took. Oh yeah, but that's like you know hurting myself. That's something. No, whatever. But yeah, so it took bio, which I was pretty experienced in, and it took dance, which I only had like an outside lens to it. I've only been an observer. I've never been a participant. And it kind of and this was actually the perfect class to combine those because it really put us not only in the shoes of a dancer, but also in the shoes of like the cell, of the DNA, of the chromosome, right down to the like itty bitty level of like each base. So it really contextualized everything in terms of movement, in terms of biology, and in terms of like molecules even.
So will you keep dancing, Stanley?
Oh yeah. You know, maybe in the shower. I'll send out like a tweet on my imaginary Twitter saying like I've been contact improving with myself, which you know doesn't make sense, but if you have a video. What? Hold on. What? I can't do this? Like, come on. Y'all some dirty people. Dirty.
As someone with very little bio experience and very little dance experience, unless it's like the Macarena or like the Cotton Eye Joe, even though I still feel like I need practice, I'm at least like a little more open to learning moves and know that I don't have two left feet in learning it, and it really is just like a step-by-step process in terms of dancing. So I'm not sure in my college career as a senior that I have time to pursue dance academically, but definitely like at friends' parties and things, I'll be more willing to learn than to sit at a table. Yeah, at the club.
Well, if there are no other questions, thank you all very much for coming.